Welcome back to topic 3.1 DNA technology. In this topic, we will go through first KPI, that is summarize the roles. So once we know that we are going to talk about the role, it means you need to say about the functions of DNA tools. For example, restriction enzymes, sticky ends and blunt ends, and gel electrophoresis. So first of all, a restriction enzyme. Restriction enzyme is the one that is known as biological scissor. So in biological scissor, that means scissor is used to cut. So whenever you see like cut, it means it's a restriction enzyme. They cut the DNA into fragments. So every cut makes two fragments. Other than that, they are very powerful tool in isolating specific regions. So how they work, let's see this one. So first of all, this is the DNA molecule. So strand number 1, strand number 2, you have the DNA molecule and you applied a restriction enzyme, and this is the restriction enzyme, the dotted lines. So it started to cut from here and it cuts until here. So when you have this zigzag line like this Z line, always make sure that it's gonna give you sticky ends. So this is going to give you sticky ends. How the sticky ends look like, they look like this. So this is how we studied. So it's going to cut from here and until here, and this is how it works. And you have two ends, and these are called as sticky ends because they have these hanging regions for the DNA cuts that are produced. So in simple language, restriction enzyme is the enzyme that recognize and bind to specific DNA and cut it. It cuts in two different ways. It cut the DNA, so it cuts the DNA into two different ways. Number one, if it cut into hanging regions, they are called as sticky ends, and they can be joined together very easily. But sometimes it cuts as this straight line. So if this is cutting as this straight line, it means it is going to be blunt end. Blunt mean rough mean, so when you need to, so you produce these two ends now. So when you want to stick this end with this end, so of course you are going to put a glue, and you know that the biological glue we studied, it's the DNA ligase. So you can join the DNA fragments by the help of the ligase, but it's not easy because if you put the glue over here and you bring them closer, they will stick to each other, but you can break them very easily. Okay, getting back, restriction enzymes are the enzymes which cut the DNA into fragments. These fragments can be sticky if they are hanging regions and blunt if they are flat at the ends. Now, moving to the next one, that's called as gel electrophoresis. But before we go to the gel electrophoresis, let's do some of the questions. Two DNA sequences were cut. So we know that they are cut by who? By a restriction enzyme. So whenever you have cut, it's very clear it's by restriction enzyme. They are cut by two different enzymes as illustrated in the diagram below. Which of the following is true regarding the diagram? So we need to look into the diagram. So enzyme 1. So I told you, when you see this cut, of course it's going to give you sticky end, and when you see this one, it's going to give you blunt. So this will result in sticky and this will result in blunt. Okay, so enzyme 1 is a restriction enzyme, yes. And fragment X is a blunt end. No, we already decided it's a sticky end, so this is wrong. Enzyme 1 is a restriction enzyme, yes, and fragment X is a sticky end fragment. So that's true. Okay, as per our prediction, that's true. Enzyme 2 is a DNA ligase. No. Enzyme 2 is a DNA ligase. No. So these are wrong statements. The diagram below shows a section. One more thing, maybe the same diagram comes but the statements change. How you can change the statements? Like this one, enzyme. So we can also say this one, enzyme 2 is a restriction enzyme and fragment X is a blunt. We can say fragment Y, so then it will be correct also. The diagram below shows a section of the DNA where two sites labeled as 1 and 2. So when you will cut at site number 1, so you produce fragment 1, and all of this as fragment 2. But now when you have another side, you cut it, so you have two more fragments. So 1, 2 and 3, three fragments. And 1 and 2, these two are restriction enzyme. Why? Because they are going to cut them. These sites are unique where the DNA can be cut. So whenever you see this cut, simply it's restriction enzyme. So don't worry about this one when you have this word that says cut. Okay, when you have this cut means restriction enzyme. So the answer is D. What does helicase do? Helicase was helix breaker. So this is used in DNA replication. Ligase is a glue which stick. So you have the keyword that is stick the ends. So stick ends. Protease is an enzyme that breaks down proteins. So protein breaking enzyme. Okay, and restriction enzyme, we know it now. Interpret the data collected from the process of the gel electrophoresis is a continuation from the previous KPI. So gel electrophoresis, first of all, we need to know it's a technique which is used to separate DNA fragments. So this technique separates the DNA fragments, and there are two conditions on which DNA fragments are separated. Number 1, charge, and number 2, size. Okay, so based on size and based on charge, it is used to separate the DNA fragments. And you put the sample over here on this end, so this end will be positive, this end will be negative. So DNA will move from sorry, I need to correct it, sorry. So this end is negative and this end is positive. So DNA will move from negative to positive. Why? Because the DNA has negative charge. DNA has negative charge. So you load the DNA towards the negative end. So negative repels negative. So it goes down towards the positive end. One more thing, as you said, based on the size. So the one you will have on the top, it will be larger in size, and one you will have at the bottom, it will be the smallest in size. Okay, so and this is based on the electrical current. You can read the remaining, and every single, like this black line, this is called as a band, and this is a DNA blend. So when the loaded gel is placed in electrophoresis tank, the electric current is turned on and the DNA fragments separate. And how they separate, we learned it already. They separate on the basis of the size and charge. Question. The image depicts the process of an agarose gel electrophoresis, in which agarose gel electrophoresis, don't worry about the agarose. Agarose is a material which is used to make this gel. So agarose gel electrophoresis is same as electrophoresis, in which the sets of the DNA size, sorry, why it stopped working. Okay, lane 1 were electrophoresed along a side sample of experimental DNA. Lane 2, so we have lane 1, which is a marker lane, okay, which is standard one, and lane 2 is your experiment. And we have the negative charge and we have the positive charge. We got 5 bands, A, B, C, D, and E, and here we have the standard band. What is the approximate size of fragment A? So fragment A is over here, okay, so it's almost 9 kb, okay. Which letter indicates the fragment that moves the fastest? Of course, the one which is the smallest, so it is E. And smaller the fragment travels the fastest through the gel, so this is what we know already. Based on the results of this analysis, did the sample likely come from any current suspect? Okay, how you know it comes from a suspect? So this is the standard DNA. Okay, you have the crime scene sample and you have suspect 1, suspect 2, suspect 3. So put the ruler. I will use my upper this black line as a ruler. Okay, so we are going to match them with this standard one. Okay, so keep an eye on the standard and keep an eye on the crime scene. So basically crime scene, let's say. Okay, so look, this sample is overlapping. Okay, this sample is overlapping here. Then I take it further down like this one, and this sample is perfectly matching with this, then I take it further down. You can use a ruler over here. This is matching with 2 and 3, this is matching with 3, this is matching with 3, this is matching with 3. I made little mistake while moving it, so this is matching with 3. So of course now it's the one that is 3. So all the bands from the crime scene, they match with 3. So yes, the unknown sample is most likely suspect 3. If you look at the crime scene sample and the suspect 3 samples of the DNA, the fragments 100% match with the suspect, so he is the answer. Use the diagram below that is obtained by the process of the gel electrophoresis to answer questions 5 and 6. So you have mother, you have father, you have child 1, child 2, child 3, child 4. Direction of the samples, movement. Which of the following is correct about the two fragments labeled as X and Y? Okay, so X is large, okay, and Y is small, and both have negative charge. Remember this one. This information you should know. Both have negative charge. X is negatively charged, yes, and it is bigger in size than Y, no. It is smaller in size than Y. X is negatively charged and smaller in size than Y, yes. X is positively charged, no, no. Okay, both of them are negatively charged. Which two children are most likely to be biological siblings and are children for father and mother show in the diagram? So the rule over here is that all the bands of the mother and father should match. So we will do the same as we did before. So I'm gonna zoom it and I will use the upper line as my ruler. Okay, so remember this is mother, so mother is matching with child 1. We go down to the father. Father is matching with 1 and 2. It's not matching with anyone else. Then the next is mother. Mother is matching with 2. So it means that who is matching 100%? Both the bands of 1 and 2 are matching, and none of the 3 and 4 are matching. So answer is 1 and 2. Sequence using diagrams and models how recombinant DNA is made. So first of all, recombinant. Re mean again. Okay, so re mean again. Combinant is taken from combine. So combine mean join. So you join the DNA once again. So what do we mean by this once again? So it's very easy. This technique helps you to make the recombinant DNA. And how you do it? You isolate a plasmid. What is a plasmid? Plasmid is a circular piece of the DNA in bacteria which is used as vector. Vector like to transfer gene from one organism to another organism. This is the role of the vector. We know DNA ligase, this is the key I told you, join or stick the fragments together, okay? Now, we have the plasmid, we isolate it, and we have the genomic DNA. You can take this from human or take it from anyone else. The step number 1, so step number 1 is isolation. So this isolation is already done. We isolated the DNA and the plasmid. Step number 2 is cleave. Cleave mean the same as cut. So cut the plasmid and the DNA with who? With the restriction enzyme. So this is step number 2. So step number 1, isolation. Step number 2 is cutting. Step number 3 is joining. This is step number 3. You join the fragments by DNA ligase, and before it was plasmid. You see all of it is blue, but now it is blue and red. From here till here, you see this is the genomic DNA that you took from here, the red one. So since it has the DNA that's not parts of its own, that's why we call it as it's a recombinant plasmid. And you will insert the last stage, stage number 4, that's called as insertion. You will insert it back into the bacteria. When you insert it back into the bacteria, that bacteria will be called as recombinant bacteria. Recombinant bacteria. So this will be called as recombinant bacteria. So make sure you know what is the difference between bacteria and a recombinant bacteria. Question: which of the following is correct? So again, we have to find the correct sequence. I told you: number one, isolation, cutting, joining, and insertion. Okay, insertion, that's over here. So they did not give isolation as the first step. It's okay. After isolation, it was cleave or cut. So cleave, cleave. So not this one, not this one. So very, very obvious. It's either A or D. So after cleavage, you join and you introduce into the host cell. Yes, that makes sense. So you introduce in the host cell, then you join. No, join comes first, then introduce comes. So this is how it works. A student wanted to prepare a recombinant DNA for research purpose. She mixed the solution that contained the gene of interest with the solution that contained the plasmid. So you got the gene, you got the plasmid, and you cut it with restriction enzyme. When she tested the presence of the recombinant DNA molecule, she did not find in the solution. Which of the following is missed? So you missed what? DNA polymerase enzyme. It is used in the PCR, not in this technique. So this is wrong. Free nucleotides, we don't need here free nucleotides. They are also used in in the PCR. Ribosomes, we use them in translation. We don't use them here. We use them in protein synthesis. So all of these three are wrong. So of course now the first is the answer. She didn't find the recombinant DNA because she did not join the cut of plasmid and the cut of the gene. Which of the following are used in the DNA cloning to transfer the recombinant plasmid into bacterial cell? So recombinant DNA into bacterial cell. So of course we use plasmids, right? Polymerase is an enzyme like the upper option as B. Primers are used in PCR. Ribosome is used in the protein synthesis, as I told you. And these two are used in PCR. The diagram below shows the steps of producing a recombinant DNA. Use the diagram. Okay, step number one, I will say here they are isolating. So step number one is isolation. Step number two is cutting or cleavage. Step number three is joining. And step number four is introducing or insertion. Whatever you can write, it's up to you. Introducing into bacteria. Okay, yeah, it's written down bacterial cell, so I will erase it. Introducing into the bacterial cell. The diagram shows an example of the recombinant DNA technology. Define recombinant DNA. So how you define recombinant DNA? Okay, so it is a DNA that results from joining two segments of DNA, and remember both of them are from two different resources or two different species or whatever, two different organisms, whatever you say. Okay, step number one, step two, step three, step four, I told you already. Better you can look for the answer key of the IPQs for these. So step one, I will just say them. Animal cell and the bacterial plasmid is isolated. Okay, so animal DNA, this is the animal DNA or gene or whatever, and this is the plasmid. They are isolated. Step number two, both of them are cut with the same restriction enzyme. Please don't skip the words. Step number three is joining. So you can say that the gene and the plasmid are joined by by DNA ligase. Remember, if you missed to write DNA ligase and restriction enzyme, you will lose half of the marks. And step number four is introducing. So recombinant plasmid is inserted into the bacterial cell. Last KPI of this topic, detail using the diagram of the steps of the PCR. So we have here three steps. Remember the three steps. Number one, denaturation at 90° to 96°, any temperature you can write. Annealing. Who anneals at this one? it's the annealing of the primers, annealing of primers at 50 degrees Celsius to 65 degrees Celsius. Any number you can write. The last one is extension at 72 degrees Celsius. Normally it is 72, but if even you write 72, 71, that 72, that will be correct. But I'm gonna erase it to stick it to 1 degree, that is 72 degree. What do you need for this one? You need DNA fragment, you need DNA polymerase, and the name PCR itself, it says polymerase chain reaction. So what is the name of the enzyme used in this one? I don't know how many of you couldn't answer in the term 2 exam. It was polymerase. The name itself says polymerase, so I don't know why it's hard to answer it. The 4 DNA nucleotides, and you need primers. Okay, now step 1, denaturation. DNA strands are separated by heating. I wrote the temperatures up. You had the DNA, you got them separate, then annealing. As the mixture cools down back to 50 to 65 degree, you attach the primer. So over here, these small segments are primers. So this one joining over here and this one joining over here, it is a primer. So you have 2 primers, they bind with each strand. Then we have the polymerase enzyme, and remember the heat resistant polymerase is a special polymerase which is called as Taq polymerase. If you just say polymerase, that is correct. Taq polymerase, that is correct. DNA polymerase, that is correct. At least say it, please. Extension, DNA polymerase extends the complementary strands by adding the specific nucleotides, because we have the free nucleotides. All these are free nucleotides, and DNA polymerase attach these free nucleotides over there. What is the end result? So end result is very obvious over here, 2. Okay, so remember that we get how many copies? 2 identical copies. The formula is 2 to the power n. So where n is number of cycles. Okay, number of cycles. So if it is the first cycle, so 2 to the power 1, so the answer is 2. How many new DNA molecules are made after first cycle? So it is 2 molecules, two copies. Use the diagram which summarizes the steps and the process of the PCR and complete the table. Okay, sometimes they say that this is PCR, sometimes they don't say, so you have to identify. Okay, step number 1, we know that. Okay, so step number 1 is denaturation. Step number 2 is annealing. And step number 3 is extension. And the details I mentioned above, you can say the details by yourself. Okay, in your own words you can also say. There is no harm to say them in your own words. So for example, denaturation. The DNA, original DNA is heated at 90 to 90. So I'm just going to say over here like this. So DNA heated, okay, at how much? 95 degrees Celsius. The hydrogen bonds break. You know that they have hydrogen bonds. So hydrogen bonds break and you get 2 separate strands. Okay, hydrogen bonds break to make 2 separate strands. What happens in the step number 2? The primers bind, right? So primers join with the template strands. Primers join, and what's the temperature at, let's say 55 degrees Celsius? Any temperature you can write, 55, 56, 57, and don't go beyond 65, and don't go below 50. Okay, so this is how it binds. And also you can say that the polymerase bind over here. And extension, who do the extension? DNA polymerase. DNA polymerase adds new nucleotides. Okay, it adds new nucleotides in step number 3 at 72 degrees Celsius to so new copies are made. Okay, so this is how you can learn. You can learn also from the and that's it, also from the answered IPQs. That's it for this topic. Inshallah, see you again with the next video. And this was topic number 2.3, sorry, it's 3.1. That's the DNA technology. Thank you.